Okay, class. I'm going to move to lecture 19. So we learned about RGD. So I'm going to switch to a presentation mode. So we learned about RGD, uh, arginine, glycine, and then aspartate. That is very important in binding to the cell. I think that is the integrin binding motif. So when cell, binds to the substratum, that is the, ECM, then it will organize the some kind of, actin fibers. One of the example in here is the stress fiber. Okay. So the circle that in the uh, figure is the cell binding site. I think that this is kind of like we call it double staining. Okay, uh, double staining that uh, red was it the green is actin, and then this focal contact area that is actually they are binding to extracellular matrix in here, right? So in here, I think that they also used a different, color of tagging protein, probably red. So that's why you will see the yellow staining over here. We're gonna see similar example a lot. So I'll just move on. So this integrin receptor is green, and then the focal adhesions are yellow because they are overlaps with this red protein that is the actin stress fiber. I think that's is the confirmation of the previous slide. So we need to go back to those properties that fibroblasts like to adhere to fibronectin. We learned, and then neurons like to adhere to laminin. So what are the things that really bring us to the cell in terms of the migration. So when cell binds to the ECM, then what will happen is these are the changes that the cells will undergo in terms of their interacting with the ECM. But the one of the important concept I want to really stress is the cell migration. So interaction of the cell with ECM is equal to cell migration. I think if I just state like this, I'm sure that I'm like a more than 80% right. Okay. So cell migration is really in other words, opposite is so true. Cell migration is interaction of the cell with the ECM. So another means that the cell will not move around if there's no ECM. Okay, right. So I think that's where that kind of notion and then where that kind of concept coming from is that we need to go back to really our early stages. This paper is published in 1976. I'm sure that none of you are born in this 1976, I guess. But this is an experiment. You have the glass microscope slide, and then you put a let's say tape around it. Okay, so you put a tape around it. Okay, something like this. Okay, right. And then on here, you spray the gold around here. I mean, it will saturate the surface of the this area. Okay, and then you after it is done, then what you do is that you remove the remove tapes. Okay, so that is the red. Then what will happen is you have the microscope slide, and then you have the area that paint coated with a gold, but also in between, isn't it? You have the area that is not coated with a gold, something like this. You have, isn't it right? So what you do is that you have a cell here. Okay, this is the cell. Okay, you have a cell, and then you spray over it. Okay, and then let them grow. Then what will happen is that some cell will drop here, some cell will go here, and then some cell will be on kind of like putting like a half on the gold surface, and then half in the very smooth glass surface. So this is what you are seeing over here. Okay, right. So you see over here, that is the smooth area. Okay, glass, so smooth area. Okay, and this area is painted with gold, so it will be considered as a rough area because it's a particle. Okay, so in the high power, it gives the surface like this. So when you see it, they are showing A, B, and C cell. And C cell is already easy, spreading over, undergoing cell shape changes. And this cell is still round because they could not move because they don't know, they don't have rough surface. And then the interesting thing is that in B, their cell body is on the smooth surface, but they are very close to this rough surface. Somehow this cell recognized, and then they are stretching their cell membrane, and try to put their feet to the rough surface. Try to get their feet wet, I guess, right? Okay, so so that process was really interesting. And then what people did is that, okay, they added cytoclasin B in there, and then they were able to block that lamellipodia. So let me just go back, okay? Okay, here. So this is the they call it as a foot. So podium. I think that podium. This is just one foot, and then this looks like a layer. So they put lamellipodia, lamellipodia, or something like that. Okay, right. So lamellipodia was able to blocked by cytoclasin B. So that is actin inhibitor, isn't it? Okay, this is the actin inhibitors. So, okay, so that is the actin dependent process, okay? So what their conclusion is that, okay, the cells like to adhere to a rough surface. I think that I would say migrating, isn't it? Cells like to adhere to a rough surface, probably that's to be exact. So they look at those, migrating cell, and then why it was able to block those lamellar, layer-like foot by the actin inhibitors. So this is the big. This is kind of like a pretty recent picture in year 2000 or something, and the leading edge. So cells are leading edge. So cells are moving this way. So you will see a lamellipodium over here, isn't it? Right. And this is a live cell picture. And then actually the lamellipodia was composed of this actin. So this is the actin stain. Okay, right. So, this is the actin, and then, that is more accumulated in this area. So lamellipodia here. So, you know, what they found is that, okay, those layer-like food is composed of this actin. Okay, right. So what they found was that, okay, you know, this is like a live picture in Nomarsky optics. And when you look at it, this is live picture. You know, you see a small, you know, actins in there. It's very specialized actin. And then when you add a cytokeratin, that is gone, isn't it? Right? You don't see just flat, smooth staining over here. So that is, you know, if you stain with an actin or something, really, the actin in here is disappeared. Okay. So, you know, those lamellipodia is composed of the actin. And then the how the cell move is most likely cell is moving around. When they move, I think that they are binding to extracellular. Not I think, okay? They are really binding to this is the ECM, okay? ECM. In here it says rough surface, but rough surface is just like artificial ECM. And then they move, and then this leading edge of area, you have a high concentration of actin. So actin polymerization at the plus end protrude the lamellipodia. So actin is organized in this way, and here is the plus end, minus end kind of thing. Okay, right. I mean, we learned about it. Okay, so as they migrate, and then also not only they are just protruding this, you know, cell membrane, but they'll also make the contact, new contact. Okay, so this new contact will contains the integrins. So not only cell is moving, but this focal contact. So, you know, touching point is also moving. Probably they will just build one new one, keep on going. And the what will happen to the other end, lagging, you know, end, is that, you know, they have a contraction going on. So you have a myosin organization and then actin, you know, different organization will occur. So cell will gain the movement like this. Okay, so let me move. Okay, so I have the this figure here. The reason why the empty, you know, slide I have is that, okay, I'm just going to draw the cell. Okay, something very similar to what we have seen. Okay, I think that's. I'll just have something like this in here. I'll just try to make the make it really more you know. Okay, and then let's say this is the my. So that is ECM, and this is the cell. Okay, so cell. So how the so cell is, let's say, moving this way. Then you know, as they move, and then they will protrude, right? And then also they will form another focal adhesion like this. Okay. So in here you will see a lot of actin. Okay. You know, pushing forwards to the plus end. And then in the focal adhesion also you have actin. So actin is somehow organized in here like this. I think that this is based on what we've seen, what we have seen previous figures, electron micrograph. Okay, right. And then the you know, here's the myosin. Okay. Okay, and then actin will be organized in here. So that's why, because they contract, so they will just be, you know, retract from here, right? And then I think that on this, this is, okay, so that is red. So red is the actin. isn't it? Okay. And then the green is the myosin. Okay, right. So, and the how they bind to ECM is that probably they're in here. There is integrin. So, you know, not just only one integrin, they have a lot of many molecule of integrin has to form here. So this is, we will say, this area, the leading edge of this area is like a lamellipodia. Okay, and then this is the lagging end, isn't it? So, you know, this from here to here is the, you know, maybe time 0, okay? And then from here to here is the, you know, maybe t equal, you know, maybe 45 minutes or something, okay? Right. So cell are moving, cell is moving this way, okay? This direction, isn't it? Cell is moving this direction. Then think about it. Cell, let's say cell has a direction and then they move a certain direction, right? Then think about it. You need to tell this cell that where they need to move, isn't it? So think about direction. And then what is giving them? Okay, what is giving them the some kind of instruction to you know, move to a certain direction to the cell. Okay, so this is the cell. I think that could be a fibroblast. Okay, right. So this is the kind of you know, overview of cell migration here, isn't it? So you need to have an extracellular matrix, and then you need to have extracellular matrix, isn't it? Okay, so extracellular matrix, and then you need to have the cytoskeleton, isn't it? Okay, so that is ready, isn't it? You have a car, and then you have a motor and everything. But the this direction thing, okay? Direction thing, we have not really talked about it yet. I think we should be able to talk about it a little bit later. Okay. So as I said, you know, it could be a video here, but I'll just skip those video. Cells are moving, you know, this direction. And then you can see they took the picture in different time point. As you can see, not only the cell is moving, but you see a new focal adhesion forms in here, isn't it? So that is a newly formed focal adhesion. So this is exactly matching with what I have talked about just one slide before. Okay, so you know, we learned you know, cell like to adhere to a rough surface, and then they are binding to you know, those rough surface. They are forming the lamellipodia, and the lamellipodia is composed of the actin, right? And then third property, I'm going to stop here, and then I'm going to talk about it in next segment. Thank you.